I'm Tuomas, and I'm working in University of Turku. You know, Leo Lahti is king of the lab that we have there. So this workshop is about the microbiome stuff. The idea is that I will present the project, and then we have some hands-on things, and then you can check the online book by yourself, and then you hopefully have an idea what is the project and can become the part of the community that we have, because we want more members in this community. It's always better to collaborate with people. This project is based on the TreeSummarizedExperiment data container. How many of you what is TreeSummarizedExperiment? Okay, great. Summarized experiment, does it say anything? Okay, how about phyloseq? Phyloseq, yes. Okay. So this is kind of phyloseq, but it's better. Yes. Yes, hopefully she did it here. But there are reasons why this is better. The phyloseq was developed for 16S data, and there are many, or not many, but multiple or several things that this TreeSummarizedExperiment solves. For instance, phyloseq, you can have only one table there. So this is designed for multi-table data, which is nowadays the case because, well, we are analyzing not only the sequencing data, but also metabolites and so forth. So we have usually multiple tables, and this is also more scalable. So when you put more samples or you have bigger studies, then phyloseq just the memory and running times just goes high, but this is more stable, so you can analyze more samples. So these are two advantages in this ecosystem. Yes, participation. We have the Alex created the virtual machine there in the Bioconductor, the Galaxy server, so we can use that. It includes all the packages that we need in this workflow workshop. And the idea is that well, this timetable doesn't hold anymore because of the problems, but we have plenty of time, so no need to rush. And I want that if you have some questions or some ideas that you want to share, just yell, because it's much more beneficial to discuss here. And of course, I can talk here as much as you want, but I'm a Finnish person, so we don't like talking too much. Yes. So first 50 minutes I will go through the basic idea of this project, and then we have the live coding things, and then you have the opportunity to go it by yourself. I briefly went through the goals of this workshop. So the idea is that you get some kind of idea what is the project, what is the ecosystem, so you can further then by yourself after this workshop you have some knowledge to go through this material, and then you can send issues or other questions to us when you know a little bit what is the project. So about the background, this will last 15 or 10 minutes. I already asked about the TreeSummarizedExperiment and these data containers, but how many of you are doing something with microbiome? Okay, well, that's great. That's great. Well, in microbiome research, we study microbes. That's the thing what we do. And the data is, well, the sequencing data creates the basis of the analysis. So what bacteria is present there. But nowadays, as I briefly mentioned, we have other omics also usually incorporated, metabolomics, proteomics, or the metagenomics, where you get also the functional annotations. And there are several data containers that are related to microbiome. First of all, there is this phyloseq. It's developed by McMurdy and Susan Holmes, and it was designed for 16S data. Then SummarizedExperiment, the core data container of Bioconductor nowadays, it was established in 2015, and then it was extended to this single cell field, and now the microbiome field with this TreeSummarizedExperiment object. And this SummarizedExperiment data container is now used in many different fields, from metabolomics to proteomics to microbiomics. And the thing is that the one advantage compared to phyloseq is that we are sharing the same data container between these different fields. So we can cross-utilize methods and collaborate. So the integration of multi-omics data, multi-table data becomes much more handy because we are now using the same methods. This is just to show you the different fields utilizing this SummarizedExperiment. So you can, first of all, you can see that this SummarizedExperiment, it has expanded a lot. So there are over 150 maybe different packages nowadays utilizing this SummarizedExperiment data container. And the microbiome is, yes, here, the pink one, it is, there are 50 or so packages. So it's a rather small area, research area utilizing this SummarizedExperiment. But if you consider all these different packages, it does not really make sense to do everything by yourself in the corner while everyone else is having a nice party and sharing methods and ideas. So it's much sensible to use the same data container. So that's the idea why this project was established. So this is the TreeSummarizedExperiment data container. And the core of this data container is these essays. And these essays are the abundance table. In rows, we have bacteria, and in columns, we have samples. And well, if we have sequencing data with counts, so each cell is how many times a specific bacteria was found in a specific sample. So that's the core of this data container. Then we have metadata for samples and the bacteria. And metadata for samples, as I mentioned, samples are the columns. So they are located in the col data slot. Then metadata for bacteria, usually the taxonomy table, it is in row data because bacteria was the rows. And this essays, row data, and call data, this is shared with SummarizedExperiment. But then microbiome or the microbes, the relations between them, they are usually represented with phylogeny tree. So we have to have a way to incorporate phylogeny, which is why Huang et al. They extended this summarized experiment with this row tree and column tree slots. Well, the phylogeny, it is related to the bacteria rows. So the row tree is the correct place to store the phylogeny. And then there is this column tree where you can incorporate sample hierarchies. So for instance, if you have relatives in the samples, there are child and parents and so forth. But this row tree is more relevant for the microbiome research. And then there is this reference sec slot, and that is the sequences. When you sequence the samples for each, you get for each row you get the sequence, so you can store this for each bacteria, for instance, the marker gene. That is the basic structure of this TreeSummarizedExperiment data container. What ideas or questions do you have on this? Yes.
So about more multi-omics. For example, I use the basis that using a HCA algorithm, I have a different sequencing for each species.
Yeah.
Maybe if you go to strain number, of course, this yes, and you can put all the reference sequence for one single species, how it works.
Yes. So each row usually it's ASV or the strain. So usually you have for each strain you have the marker gene or sequence that is related to this specific strain. So then you can map these different sequences to that strain or row. Did that answer to your seems to?
Because I think that this can be bigger.
Yeah, but it's this structure itself, it's very flexible. So there are different ways to use this or utilize this structure. So it's well, yeah. These are just what I'm talking here. These are how we are using this TreeSummarizedExperiment. This is more generic data content. You can use any kind of this kind of hierarchical data that you have. So it can be also utilized broader outside of the microbiome field. Yeah, what other thoughts do you have?
Right, that row tree is used for taxonomic relationships.
Yes, so you have the phylogenetic tree, so you can put it in the row tree. So rows are bacteria, so row tree is the place. Yes, good.
And so, could you just clarify what row links?
No.
How do row links differ from?
Yes. Sorry about that. Yeah, I missed that completely. Very good question. So in the row tree, we can have the full phylogeny tree. So this row link, what it does is that it linkages these nodes from this tree with these rows in this TreeSummarizedExperiment data structure. So for instance, you can see that these rows, they are orange color. So these rows, this and these rows, they are mapped with this node in the phylogeny. So they are mapped with the same node in the phylogeny tree. Oh yes, so future people are also see. Then we have this, whatever color this is. So this row is mapped with this node in the phylogeny tree.
So not every feature in your SummarizedExperiment is represented individually in the row tree.
Well, it can be so, but because this is very flexible structure, so it just happens so that this node is, for instance, if these are these rows are strains, and this phylogen is species level. So it could be that two strains are linked with the same species. So that's what happens here. So these rows are related to this node in the phylogen. Good. Yes.
Very nice.
Yes, so usually it's you can get from the database if you're utilizing the metaflan, for instance. Then in the database you get the phylogeny, and it's usually the Newick whatever tree format it was, and then you can just import it this data structure. In the online book there is examples on how to do that. Good. Yes. But it's very useful to go through this data structure, because that's the core, and if you don't know what is the data structure, then you cannot really utilize these methods. Very good discussion. Yeah, so that's the data structure that we are using, and well, the data structure without the methods is like a car without the engine. So what we have done, we have created the engine. So there are different methods that we have developed. There is MIA, the core package. Then there's a MIA Vis or visualization, MIA Sim, MIA Time, and MIA Dash and IC3 for the graphical user interface. But the thing is that this is not only our project. It's this community project, and it's very nice that now there's more and more people that are supporting this ecosystem. So they have developed methods. For instance, there is this Anansi package. So that they have developed packages that are supporting now this TreeSummarizedExperiment ecosystem, which is pretty cool. And as a cherry on top of the cake, well, this TreeSummarizedExperiment is summarized experiment, so we can cross-utilize these methods from this broader summarized experiment ecosystem. Cool. What thoughts do you have on this background that I discussed? Cool. Great. So then we can start this demonstration. So there is this workshop.bioconductor.org, and I'll probably use this one. Yes. So what I want you to do is that go to this website. Does everyone know what is the website address? It's pretty small. Well, I cannot make it bigger. Everything's good. Yes. So there is this Orchestrating Microbiome Analysis with Bioconductor. So click that and then run tool, so you can get the virtual machine going now. And it's running. So then please press that and it opens you the RStudio. Yes. How many of you were able to? Is there still someone that? Okay. Did you find the address? Okay, and it's still compiling the starting the thing.
Yeah, I will.
No. It's much better. Do you have the address?
No.
Yes. No.
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 I will put it
Oh, yeah, because I don't have that.
Workshop done by Bioconductor.
Yes.
Single.
Yes. And then there was this, here it was if you go to homepage, you can see there's workshops and then this Orchestrating Microbiome Analysis with Bioconductor.
Yeah, but they count. They asked me to.
Yes, yes, there you must have account, yes.
I can register. You can register, but not.
Okay.
You will register.
Yes. Yes. So I can a little bit tell you already about the dataset that we are going to use to weight people. So there are, in addition to methods, data structure, and other things, there is this openly available databases. That's a pretty nice thing. And these databases, you can easily get the data from there because you can use these dedicated packages that fetch the data directly to TreeSummarizedExperiment object. And there is, for instance, this created metagenomic data by Levi-Waldron lab. And there is, I guess, 100 created metagenomic datasets. And well, you can of course utilize this to get new insights from the data, but also if you have developed method, you can benchmark your method with these real life datasets. So this is very handy. Then there is Magnify. How many of you know the Magnify database? So it's a very large European Bioinformatics Institute database. I guess over like 500,000 samples there. And there is Magnify R package where you can fetch the data from Magnify directly to TreeSummarizedExperiment object.
And so the data is quantification of it's all already reprocessed.
Yes, and the thing is that they are using the same pre-processing steps for each sample. So it should be comparable. Of course, there are different technical things, but in theory, they should be comparable. Yes. Are we now ready to?
Their studio, but
Okay.
not to.
Yeah. So. Is it working your RStudio? Yeah. Okay, cool. So we can start now the demonstration. So there was this created metagenomic data base. So we can to fetch data from there is this created metagenomic data package. It was developed by the Levi-Waldron lab, and with that you can fetch the data directly to the TreeSummarizedExperiment object. It seems that it takes little, yes. And it was this command created metagenomic data. And then from this package, there is function called surprise created metagenomic data. So we can utilize that function created metagenomic data. And what it wants is the pattern of the study. Well, we can check from I will check here quickly because I don't remember by heart. Oh, this is wrong. Where is it? Sorry about this hassle. Was it this? Yes. So it was the pattern, and it is this pattern. The first part of the pattern says the study, and then this dot relative abundance. It's the definition of the taxonomy mapping of the data. Yeah, these are metagenomic samples, so there are also functional annotations. Then what we want is that we want try run equals to true, because if it's no equals to false, because if the try run is true, then it just fetches the available datasets. So we want dry run is false. And then what we want is the row names equals to short. It just makes the row names nicer. And we put this data to TSC variable TreeSummarizedExperiment. And now it asks if it's okay to create the cache directory, and it is okay, so we say yes. And it shouldn't take too long. Just yes, now it's ready. And what it gives us this list. So TSC is now a list of TreeSummarizedExperiment objects. So that's why we get the first element of that list. It is our dataset, and we put it in TSC variable. And now we can print the TreeSummarizedExperiment and check how it looks like in our console. So first of all, it says that it's a TreeSummarizedExperiment. That's the first hint that we have the correct data. And as I didn't actually mention too well how it works, but we have the rows. This works similarly to any like rectangular data class in R. So we have rows and we have columns. So we can subset this data. This is rows and this is columns. So we can, for instance, subset the data very similar to matrices or the data frames in R. So for instance, we can get the first column, first sample, and the first 10 rows, and it looks like this. And then I mentioned that there is this, I will print the original dataset. So I mentioned that there is this essays is the core of this data container, and it contains the abundance table. So here we have the essays, and it says that it's the relative abundance. And to get the essays, the abundance table, we can use the essay function. So essay, then TSE, and then we have to specify the name of the essay, which is the relative abundance. And it gives us the abundance matrix. Then we have the row data there, which is what was the row data? Anyone? What is the row data? What data there can be in the row data slot?
Prior to relative abundance?
Yes, there is this essays is the core of this data structure. Then we have metadata. So there can be metadata for rows, which are the bacteria. Yes, and then we have the columns, which are the yes. So for both these, we can have the metadata, and row data is the metadata for row, and then call data is metadata for the columns. Yes. So we can check the row data first. It was the taxonomy table, and it looks like this. And then we can print the sample metadata with this call data function, and it looks like this. So this is some colorectal cancer study. I guess, yes, there is this disease type is the colorectal cancer. There is also healthy ones in this data set. And then we can check the row tree with this row tree function, row tree and then TSC there, and it looks like this. So it's just a representation of this phylogenetic tree or row tree. There are functions to visualize this. This does not tell us much, but it is present there. Yes. What thoughts, questions do you have on this?
So just to clarify, if you subset species, you subset row links that I get, and the row tree is immutable, it just doesn't change.
Yes.
You don't lose like the leaves that you filtered out.
Yes. Yes. So when you subset, for instance, so that you take first 10 rows, so bacteria, what it does is that, let's put it in TSC sub. What it does is that it subsets the abundance table and the taxonomy table. So we can check so that I don't lie. TSC sub. So there is 10 rows in the taxonomy table. And also what it does is that it subset the row links. So you can see that there is 10 rows. But what it does not do is that it does not do anything about the tree. So it's intact, it's still the same.
So it doesn't prune the tree.
Yes, it does not do that. But you can do that manually after when you have subsetted the data. Yes, great. Other thoughts? All right. So then we can continue with this. Well, first thing usually is that we want to agglomerate the data or aggregate the data. How many of you know what is the data agglomeration in this? Okay, cool. Yes. So what it does is that we have, for instance, I can show you the row data again. So for instance, if we aggregate the data to phyla level, there is phylum. So what it does is that it for each this row is agglomerated so that these bacteroidata, this one, this one, this one, and this one, and also this one and this one. So each this bacteroidata, they are summed together. And then there is this firmicutes 2 times, so these are summed together. So that's the what agglomeration does. And why it is usually done? Well, there can be different reasons. For instance, to get biological more meaningful features. If we do, for instance, 16S sequencing, usually it is said that the species or ASV level, it does not say much because the genus level is the lowest level that you can identify features. So that's one reason. Or then it can be just to if you do a statistical test So to reduce the number of tests, you can agglomerate the data and for instance to phylum level or class level or so forth. And this is very basic operation in microbiome field, and how it is done is that we put the TSC in the same variable. We overwrite this variable by running the agglomerate by ranks function. And when we do this, happens that. And why that happens? Yes. So to load we load the MIA package. It has the common operations for microbiome data science. So now when we run the same thing, TSC and agglomerate by ranks, then we can print the results soon. It will take a couple seconds, and then we can print it again. It seems that nothing happened. So we have still here over 300 rows and 60 samples. But what it does is that it agglomerates the data to each taxonomy level and puts this agglomerated data to this alternative experiment slot. So you can see that this alt-exp names is now populated. It just puts the data there in the same data container. And why it's useful when then you have this data container does the bookkeeping for you. And this is something that you cannot do with the phyloseq. So you can have only one table there, but this can handle this, for instance, agglomerated data. And when we subset for instance, samples, it subset all these different these agglomerated data in this alternative experience slot. So that's very convenient. And then we can check, for instance, the phylum level here with alt-exp function. What it wants is the TSE and then the alternative experiment that we want to fetch. And it is similar object, but what you can see is that now it has 11 features. So there is only 11 phyla in this dataset. And of course, the number of samples is still 60. Then we can check the alt-exp, the phylum level, the row data of the phylum level data. We can put this inside row data function. And now it looks like this. Is there something problem with the data? Why there's so many missing values? Yes. So that's why there are. This is what happens. This Actinobacteria, for instance, we have lost all the lower level information. So that's what happens when you agglomerate the data. Cool.
Tuomas, is that expected that when you switch up into experiments, you lose the other ones?
You switch, sorry.
If you check out X7,
Yes.
then you lost the others.
No. So you mean that if I do this and then there is nothing?
Yeah.
Okay. Yes. Cool. So this how this done? This is the TSC, the main object. So if we check that, then we have in the alternative experiment we have the different experiments. So,
yeah, but
yes.
Shouldn't you be able to like switch and then between the different alternative experiments?
You can do that, for instance, if you want the genus level, it's the same command.
Okay, I know. It's just
So they are.
When you switch to an other alternative experiment, you keep the other ones.
Yes. It's hard to put in the words, but what it does is that these all alternative experiment, genus, phylum, kingdom, and other levels, they are stored in this main tree-summarized experiment. And what they are, these different alternative experiment, they are tree-summarized experiment objects. So they are like stored, these tree-summarized experiment are stored inside the main tree-summarized experiment.
Yeah.
And that's why if you, like, for instance, print the genus level data, there is nothing in the alternative experiment, but the main tree-summarized experiment has these alternative experiments. So that's that's why it's there, because they are stored in the main data container, these individual agglomerate data. Yes.
We used to use the merge feature by rank function.
Yes.
Just
Okay.
that
Yes.
going to be duplicated. And previously we used to create new TSC objects, genus TSC, species TSC,
yes,
doing subsetting. So now what you're saying is that it will all be stored inside the same TSC.
Yes. So there is still this agglomerate by rank function. So you can see there is rank and ranks. And with this rank, you can agglomerate the data to your some specific level. Yes.
We would store in a new TSC object.
Yes. But it's might be more convenient to use the alternative experiment, because that does the bookkeeping for you. Then you have only one object that you manage.
Then back.
Yeah.
So I figured out if you use the swap out X function,
Swap.
that does the swapping. It's
okay.
actually super convenient.
Okay.
If you lose the other ones, it actually swaps the main and alternative.
Okay. How well? This first time I see this.
The main purpose of having alternative experiments is to be able to swap between.
Okay. Yeah. Yes, very nice. It's actually very powerful, because as you can see, it's the single cell experiment. So it's because we are using the same data container, we can cross-utilize these methods. And there are lots of different methods, and it seems that I have to still read before my bedtime these different methods.
Because that allows you to operate on at one rank and then switch to another rank and then do further operations, and just like iteratively switch.
Okay, yeah. Okay, that's very cool. Thanks. Great. What other thoughts on this? Yes. Yeah, lots of work.
Thinking from the point of view of the end user, like it would be an epidemiologist that we have in our group, that the way you are thinking of storing everything inside the same object that has the same object, it's I understand why you prefer this way, because it's more like a software engineer way to do it.
Yeah.
But for anyone that doesn't have this background, I think it's more complicated to them to understand this logic of working.
Yeah. And yes, when you use the alternative experiment, it complicates things, because it's then a much more complicated structure. Yeah, I can understand that point of view, but yeah, it's as I said, this is very generic ecosystem. You can do whatever you want with this. It's like we just give you the tools, and then you can build a house or whatever with this. Yeah. Great.
All these agglomeration and things, is this function the one that has the option on rank only that then would erase the missings or keep account of the missings and also enumerate the missings? Is this the one?
Yes, there is this functionality. Yes, the on rank only. I cannot remember by heart now, but there is in the documentation. Yes, so there is this option.
Ask if you would think it's better to use on rank only as true or false, depends on the context. Because you are losing information at the end, because sometimes you have a missing in a specific rank, taxonomy rank, but it's because it's an unassigned genus from a known order or from a known class, whatever. So it's information that you are missing. Yeah,
that's true. Well, I don't know. It's maybe more biological question. I don't know. But yeah, what it does is that when you have missing data there, then if it's on rank only, if that is true, then it throws away all the rows that does not have the information in that specific level, for instance, genus level. And if it's a false, then it finds the highest, the lowest available level. For instance, if there is only family level, then it assigns it to there. But yeah, there are different ways to do things, and unfortunately, I might not have answer to that. Yeah, great.
And just out of curiosity, am I right understanding? So I tried, silly me, I replaced genus by one across the whole column. I did that. Try, and then when I agglomerate by ranks, I still have 3 genuses.
Yeah.
So my right understanding that it uses the whole tree to do the agglomerate, not like starting with super kingdom, and then within is 3 different super kingdoms. It found like a group of 1, and a group of 1 in genus. Does that make sense?
I did not get exactly
So
that point. Sorry.
I put 1 across the whole genus. Column.
Yes.
And then did agglomerate by ranks. So I was expecting just 1.
Oh yeah. Yes. Okay. Yeah.
The genus.
Yes.
But I have 3, and I assume that's because prior to genus rank is like the 3 super kingdoms.
Yes. Yes. So there is this agglomerate by rank function, what we have now used, and more generic like agglomerate by variable. And with agglomerate by variable, you will get the 1. But then there might be that in the database there is the same name for genus, but they are assigned to different families. And this agglomerate by rank does that take that into account. So that's why you should use when you agglomerate the data based on the taxonomical ranks, you should use the agglomerate by rank. Great. Then we usually transform the data. That's the very typical. There is different transformation methods in MIA package. There is more information in the online book, but well, for instance, the relative transformation is one of those typical things. So we can do that. How to do that is we have the TSC variable, so we will overwrite that and run the transform essay function. And what it wants is the TSC object. Then it wants the essay type that we want to which we want to apply this transformation. And well, there is this relative abundance table already, but we will apply the relative abundance as the relative abundance, if that makes any sense. But we will now do that. So it's relative abundance is the essay in the essay slot. And then we want to apply the relative abundance transformation, which is the rel abundance. And what it does is that it adds new assay to this assay slot. So now there is 2 assays. And that's something that you cannot do with phyloseq. That's it every time. That's what it does. And then, of course, we can apply robust CLR, center to log ratio transformation, and the method is RCLR. And the same thing, it adds now there is 3 assays. And again, that's not something that you can stop that too much. The phyloseq is quite very nice. There is a huge ecosystem in phyloseq, and that's why it's something that people are still using. And it's very very big community, which makes it very nice. But back to the TreeSummarizedExperiment. So yeah, so it adds this transform table in the same data structure. Then we can visualize this. One thing, sorry. So what it does is that it applies only these transformations to our main data container. But if we, for instance, check the alt-exp and the phylum level, you can see alt-exp function and TSC and the phylum. You can see that it's not applied to this agglomerated data, but there is option also to, there's alt-exp, and then you can put alt-exp names, which is the vector of each these different alternative experiments in the data container. And what it does is that it applies these transformations to, for instance, the phylum level. And now you can see that there is this robust CLR transformation. And I will do the same with the relative abundance to get things harmonized. So the same command, but there is this now the rel abundance method. Then we can, for instance, visualize this. There is the MiaWiz library, and it has the visualization. Yes. Yes. Yeah.
We can add, say, applying some function on the say that we have a custom function. For example, I want to have the count of pseudo count from the relative abundance. I can multiply everything for 1 million, because after that I need to use some counts for statistical analysis that and because what I do by myself, I import from a table. I export a table as a say. I calculate from a table, and after that I add the say from external. But there's a method to use, for example, let's say relative abundance and multiply for 1 million.
Yes. So this is just a core structure, but you can always take the abundance tables, for instance, out of the structure.
Don't take that out.
Yes, but then if you take it out and then do the customization, put it back.
Yeah.
So yes. Okay. So there is no that kind of like a transform essay with your own method, but what you can do is that you can TSC and then take the relative abundance. This was the raw values. Take that, and then we can multiply it 1000, and then we can put it back into the essay with our transformation like this, and it probably, yes, it works. And now we can check that there's our transformation.
Only for that main experiment, not the other experiment.
Yes, that what I did does not it does not apply the transformation, but you can do it manually. Yes. Cool.
It could be a nice feature to add. Yeah, to have some within transform assay you can provide a function.
Yeah. Yeah, it could be.
Like or method you take a string or function.
Yeah. Yeah, that's a good idea. But on the other hand, the idea is to standardize how the people do the microbiome research. And currently there is a quite comprehensive set of different methods that are commonly applied in the microbiome field. But yeah, this is good idea, and it can be implemented. We have to discuss.
Something that is very difficult here.
Yeah.
When you use the metaplan data.
Yeah.
You at the length of the reads, the number of the reads that you enter in the plan,
Yeah.
and you multiply for this relative number, the counts, the physical counts that you have, you compare to some particular statistic method that you apply. That will need to the counts, the counts.
Yeah.
So something that I use day by day, so maybe it can be used.
Okay. Well, good to know.
And also, I think it would. So I don't know the internals, but because I see that you have a number of different methods.
Yeah.
That you can apply. So if you have a generic one, then you can easily implement new.
Yeah, that's
methods.
Yeah.
Or like because you have a number of them. If you figure out that there's a new one that you believe should become one of the standards, then
Yeah.
it becomes easy to add on top.
Yeah. Yeah. It shouldn't be too big thing to implement this kind of thing. Yeah, that's a good idea. Yeah, we will discuss this and probably we can implement that. Great. That's very nice to get new ideas, because we don't know how people are actually using this, so it's very nice to hear how people are actually using these tools, and I get new ideas from that. Yes. So what we can now do is that we can, for instance, visualize the relative abundances in phylum level, for instance. So it's the library. MiaVis is the package for visualization, and there is this plot abundance function, and that creates the nice bar plots that you can see in many publications. And here we want the alt-exp and TSC phylum. This is the phylum level data. Then what we want is that we want to visualize the relative abundances. So TSC phylum, and then it wants the assay type, and it is the rel abundance, like this. And what it does is that it creates this kind of bar plot that you can see almost every publication. It's a very common thing to visualize this, for instance, phylum or top 20 genera or something like that. And then we can add there, because this bar plot does not show us much yet, but we can visualize the column metadata. So we can first check the call data, TSC, and we will see that there is this column named was it? Yes, disease. So we can visualize this in our bar plot. How to do that is the same command, but then there is call var argument, and if we put it disease there, it will create it as a list of plots. But there is better, it's a list of plots. The first one is the same bar plot, and the second one is this kind of additional plot, and we can then wrap this. But there is a better way to do that. It's the facet calls, and what it does is that it uses the ggplot functionality to create this kind of panels in the plot. And now it looks like this. So there is now there is this sample information here. I can see people are seeing the white space here. So that's because this is used a fixed scale in the faceting this panels. So we can use the different scale in the scales and 3x. And now it looks much better. So this is one way to visualize this the phylum level taxa, and we can already see it's not too usual that you will see this kind of very raw summaries, these kind of differences. But in this dataset, you can see that there is proteobacteria is more present in the colorectal cancer. It's this color here. What thoughts do you have on this? Yeah. Then one very typical thing is to calculate alpha diversity. And alpha diversity is the metric which tells how diverse the microbial community is. And what those alpha diversity metrics, what they do, they consider how many different bacteria there are in a sample and how evenly the abundances are distributed. And then there are different matrices that weights these two aspects a little bit differently. And there is a function called add alpha in MIA package, and what it does is that there is an error, and it's because it's wants the assay type, so we have to specify the assay type. That's the error. So then we use the relative abundance, which was the raw data. Now it should work, and it works. What it does is that it adds these alpha diversity indices to our sample metadata. And each sample, for instance, there is this Shannon diversity. So each sample has unique value, which tells how diverse the microbial community is in that sample. And then we can visualize, for instance, this Shannon diversity. We can visualize that. And there is a function. We can visualize that, for instance, with box plot. So there is a function called plot box plot. And what it wants is the TSC. Then it wants the column variable with call var argument that we want to visualize. It was the Shannon diversity, and we can print that, and it gives us the box plot that shows the distribution of the Shannon diversity in this data set. But then it's useful to compare these colorectal cancer to the healthy ones, so we can add the x-axis, and it was the disease, and it looks like this. So you can see from here that the healthy ones have lower diversity compared to the colorectal cancer patients. Yes.
This will test on this, or?
Yes, there is a way. There is an online book. There is an example how to, for instance, run. Yes, you can also do that. But there is a pull request in the MiaWiz package that the pull request will add the functionality to this plot box plot function, where you can add the p-values. For instance, here you can compare with Wilcoxon test if there's if the difference is significant. So yeah, it is also possible to add new layer with this GG. What was the? Well, there are different packages that you can add. Yeah.
So I need to see if I need to use a packet or
Yeah. Well, we have something in like very soon.
Yeah.
Yes. 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 Oh, cool. Yes. Thank you. Then one thing that is also we already did the alpha diversity thing. So, but then we usually do the beta diversity, or for instance, ordination, principal coordinate analysis, for instance. And how many of you know what is the principal coordinate analysis? Yes. Yes, so it's kind of like a principal component analysis, but now we can specify the dissimilarity matrix or the dissimilarity metric. So principal component analysis is Euclidean distance, but usually we want in microbiome field we want to apply, for instance, Bray-Curtis dissimilarity, so we can do that, but then it's called principal coordinate analysis. Very similar, but the name only differs. So we can do that, and there is this function called add MDS, YMDS. So it's the multi dimensional scaling. Yes, cool. We agree. And what it wants is the TSC, the TreeSummarizedExperiment, and then assay type, which was the relative abundance. It is common choice to apply this Bray-Curtis with relative abundance. There are a couple standard choices that you can do, and you will find from the online book more about this. So for instance, with the CLR center log ratio transformation or the transform data, you can then apply the Euclidean distances, the principal component analysis. So there are different standard choices, and the relative abundances with the Bray-Curtis dissimilarity is one of those standards. So what it wants is the TreeSummarizedExperiment. Then you have to specify the assay where we want to apply the method, and then it wants the method, which is the Bray. It's the Bray-Curtis, and then what it does is that it adds the results to this reduced dimensionality slot. So we can see that there is now MDS. And you can of course, fetch the data from this lot with this reduced dim function, and then it wants the MDS, which is the name, and this is the result. So you have here two coordinates or components, and there is this samples now in the row. But this results itself, they don't tell us much, because usually we want to visualize this. And there is a function called plot reduced dim. Yes, I cannot find it because it comes from the scatter package. It's the package from single cell analysis world. So we can cross-utilize these methods, and there is this plot reduce dim function. What it was is once is the TreeSummarizedExperiment object, and then the name of the reduced dimensionality results, which was the MDS. And what it does is this scatter plot. So we can already see that we can already see that there is some differences in the data set, but usually we want to color these so we can see if there is a difference compared to these study groups. And how to do that? It's the color by argument, and it's the disease. And it looks like this. So it seems that there is association between there is, for instance, here is the healthy ones. There's only like quite nicely clustered healthy ones here, and then there is in the middle, you can see that they are kind of mixed, but there is these colorectal cancers patients here, and well, how to interpret this? If you want, you can see there are usually patterns, and well, I want to see patterns, so I can see that there is some association at least. But then, of course, you can apply statistical tests to confirm that what you see in this ordination plot is actually also statistically proved. So there are different methods you can run, for instance, Bermanova or the redundancy analysis. And these both are in the online book, where you can find more information. Yes, what thoughts do you have on this? Yes.
So, visualization of this plot. In this case, there are two categories of the disease. If, for example, I have three or four.
Yes.
How can I, for example, add an ellipsis circle to visualize this pattern?
Yes. In this plot reduced dim, there is no functionality, but you can always add their layers, but I don't know how well they work. But there is a pull request also on that in MiaWiz, and with that function, it's called plot ordination. It's not yet there in MiaWiz package. It will be there soon. We are adding their functionality to add ellipses on that grouping. Yes.
I use the same function that I export people of the.
Yeah.
Then I got layers.
Yeah.
You can add ellipses, you can add the bottom, the borders, you can add everything.
Yeah.
I can send you to the code on the GitHub.
Okay. Yeah. Cool. Yeah. Yeah, it is because we are using ggplot. It's you can always add layers there. It's convenient. other thoughts? All right. So that was the live demonstration. Because of the hassle, there's not much time for you to do it on your own. But what I still want to show, there's 9 minutes left. I want to show you the online book, because that's something that very useful, hopefully in the future also. So there is this, the address is this, microbiome.github.io and then OMA. And you can find from that address this online book. From this online book, there is different chapters, and the idea is to show the practical examples how to manage and analyze the data in this TreeSummarizedExperiment ecosystem, but also to show best practices. So, for instance, if we go to the diversity, the orientation stuff here, so you can see that there is a table of standard ways. So the standard the transformation and beta diversity metric. So for instance, if you are applying Euclidean distances to center log ratio, it is called Aitchison distance, and that's one of the standard choices. Or then if you are applying the Bray-Curtis the relative abundance, that is also one of those standard choices. But of course, these are not these best practices are not formed by our group, but it's formed by the community. So that's why I welcome each of you to join this community and create issues, ask questions, and do pull requests and involved in this community, because it's very important that there's more than just one group doing this stuff. Yes, of course, these what they are the best practices that we are now listing here. You might disagree, and then we can discuss. But there is always, when we say something that this is a good thing to do, this is standard way to do, there is a citation that shows that why this is a good way to do. So we have not just created these best practices, but this is based on the community and the publications. Great. This was that. I will put the address here, still the OMA online book. What thoughts, ideas, or questions do you have? You probably have many of them. So please.
Suggestion about the statistical and the different abundance analysis.
Yeah.
Because there is tons of approach that are used in the field usually. And if you have a relation with the different kind of data, if you have right, for example, metaplan respect other stuff. I mean, Maslin or AMCOM or ALDEX or so if you have some suggestions.
Yes. Well, as I mentioned, there is in the online book there is more information, and this is now updated. This is the latest one. It was updated today, and there is, for instance, this Juho Pelto and others, where they compared different differential abundance methods. And what they found was that these elementary methods, Wilcoxon test or t-test or the linear models, you can always model the Wilcoxon test and t-test as a linear model, but in linear models you can add covariates. So these very simple methods, they are working better than these other approaches. And Marsin it's one of the it is using linear models under the hood. So that's the Marsin 2. It's one of those methods that I can suggest to use. But then we have a project where we will develop a dedicated package for differential abundance methods that are supporting these elementary methods that this publication is mentioning. So linear models and with relative abundances and then there was this other. You can check the publication. I really that it's very good publication, and there are also other publications that have found the same thing, that the elementary methods are the best ones. But yeah, in the future there will be a package that includes all these elementary methods and the visualization. So
you're suggesting just one method, or to try different one and see if there is some small tools, some systems that are coming up from the different, because I read a lot of articles.
Yeah.
And there is two strategies. One is to choose, for example, for Metaplan must be because are developed for people on Metaplan.
Yeah.
Maybe they have the data down there, or to use a couple of methods, 3 methods, and say, okay, I have this method, this method, this method. There is also a package in Bioconductor that do this automatically.
Yeah.
And they put together and say, okay, this one is this piece.
Yeah, it's the bench-dummy package, and it's supporting the TreeSummarizedExperiment. So you can easily apply these different methods. But I guess this is more like philosophical question, because usually it leads, if you apply all the methods, usually it leads in that situation where you are like cherry picking that are favoring your yeah. So I guess the best would be to first choose the method and then apply the method, and that's it. But usually it's not. It's usually iterative process research, so it does not go that way usually. But if you're using these elementary methods or the standard methods, it's usually safe. Very good. What other questions? All right. There is still 1 minute.
Okay.
Okay.
For functional data, for example, data from human or functional stuff, you can integrate directly on the object, or you need to use, for example, 2 different objects, one for the functional.
Yes, so you can edit, for instance, the alternative experiment slot if you have one-to-one mapping, and you have usually when you are the human pipeline, for instance, you get the functional annotations and the taxonomic annotations. So it is one-to-one mapping. So you can use the alternative experiment. But if that is the case that you have metabolomics data, for instance, there is some samples missing, then there is multi-assay experiment, which is more capable data structure. And there is examples on that also in online book.